All right, so it's my pleasure to introduce Andres Cardenas. He's an assistant professor in the Department of Epi and Population Health at Stanford School of Medicine. Prior to that, he received his BS in biochemistry and biophysics, his MPH in biostatistics, and his PhD in public health with a concentration of environmental health. He did his postdoc at Harvard University with Andrea Beccarelli, and I think that's maybe where you got a lot of your epigenetic experience. Before joining Stanford, he was an assistant professor in residence at UC Berkeley. So he loves the East Coast and the West Coast. I don't know, you're in the middle of the US, or you're surviving?
Things, yeah.
Okay. He's certainly a clear leader in environmental epigenetics and epidemiology. As an assistant professor, he currently is the PI of 2 R01s. One of them is a ONES Award from the NIEHS. He's also a PI of an R21 and is also a program leader for the data management core of a Superfund P42 NIEHS grant. Assistant professor, he has 132 publications. I mean, this is really remarkable, more publications than I have, so kudos to that. What else? You're a co-I of another 5 R01s, and he obviously have many accolades. The one I'm going to mention in 2020 is recognized as the Outstanding New Environmental Scientist Award for NIEHS. So I'll let you take it away.
Thank you so much, Rick. So, thank you. Yeah. Well, I'm really excited to be here today. I really appreciate the invitation, and it's been really nice meeting with people and getting to know Wayne State. I have to say, I'm really impressed. I mean, you have the P30, a P42, and there's a lot of really exciting science happening right here, right now. So congrats to the amazing work of the entire team here. So today I'm going to be talking about epigenetics and its relationship to health and disease. As a disclaimer, I'm an environmental epidemiologist, so I mostly do human studies. I have collaborated with toxicologists in the past, but given that there's a broad audience here of epidemiologists, molecular folks, toxicologists, I'm going to try to give you a really brief overview of the work that I do and some of the concepts that I think are going to be pretty key into the future as well. So I don't know if we're getting a little bit of feedback. Are they going to try to lower the volume? Okay, yeah. Okay, great. So when I talk about epigenetics, I like to introduce the central dogma of molecular biology. This might be a little bit outdated, but I think most biologists will agree that this is how we think about living systems, where DNA can replicate itself to make more DNA. This is fairly static for all practical purposes, right? You're born with your genetics, and for all practical purposes, things don't change over time, right? So it doesn't matter if you get your genetic sequence once you're born, or if you get it when you're 80 or 90, it's going to be exactly the same. There's errors that happen. There's systems in the cells that fix these errors. But for all practical purposes, and for the population level genetics that we do, where we learn about genetic diseases, genetics tends to be fairly static for the most part. Of course, there's CRISPR and Cas9 systems now that are very exciting to try to solve some of the challenges right now. But for the time being, genetics is static, at least the way we understand living systems. There's this transcription process where you go from DNA to RNA. That's less stable, right? So RNA can be degraded in a matter of hours, and it's time dependent too. So if you think about this, you have the instructions of life coded in the DNA. That's your blueprint. Then you're passing on a message of RNA, which is very important to try to activate pathways and things. And then finally, you get the molecules that actually go and do things, which are the proteins in the process of translation. So you have this kind of layer approach to try to regulate the flow of information from a blueprint that is fairly stable, DNA, not changeable, to transcription, where things are somewhat stable, to proteins, where things are a little bit more unstable over time. And as you can imagine, this is good because you need to activate different genes at different points in time. So this is what I'm going to introduce, another layer of complexity to this molecular view, which is epigenetics. Epi comes from the Greek above, genetics, so it literally means above genetics. So these are changes in gene expression that do not depend on the DNA sequence itself. So you're not changing the sequence, yet you are altering the expression of said genes. When I say stable, I mean mitotically stable. So from one cell to another within an organism. I had the opportunity to discuss with some of you transgenerational inheritance. This is highly debated in humans, even in mammals, although Rick has done amazing work showing that it is possible maybe to epigenetically transmit information from one generation to another. In mice, the data in humans is not that compelling. So this topic is highly debated. But if you talk to nematode folks or people that do experiments with plants, they see this all the time, where there's flow of information that is epigenetically passed on from one organism, from one generation to another in a non-genetic matter. So if you take anything away from this talk today, is that epigenetics is tissue specific. So you can see there the process of differentiation from the zygote to forming the different layers of the gastrula. And very early during development, if you think about going from this organism that only has one sequence of DNA, so all your cells in your body, doesn't matter if you sequence your vocal cells or if you sequence your blood, which are leukocytes, they basically have the same genetic sequence for the most part, for practical purposes. Yet they have very different functions, shapes of cells, different organs that they form, and this is through epigenetic programming. So early during development, the cells differentiate into these pluripotent stem cells that might commit to become different layers of organs, and this is to epigenetic programming of cells, and I'll talk a little bit about that. So if you take anything away from today's talk, is that each cell type might potentially have a unique epigenetic signature. So you can imagine the level of complexity when you're trying to do this. Even in experiments, you have to think, okay, I'm not just sequencing DNA, where you get the sequence from blood, usually has plenty of DNA, but you have to think about what organ is your target organ to try to get at that. Very similar to gene expression, where gene expression is tissue specific and cell type specific. Here we have the complexity of cell type specificity, but I also think it's an opportunity, and I'll talk about that in a little bit too. So the NIH realized this is probably pretty important to know about these epigenotypes across tissues and really try to understand disease susceptibility. So early in the 2000s, we thought that we were going to have a genetic marker for every single disease and even complex diseases. But we're beginning to realize that even now that we have whole genome sequences among humans, it's very difficult to explain disease susceptibility with genetics alone. There's a few exceptions, right? There's diseases that have a high genetic penetrance, but for the most part, complex diseases, things like diabetes, cardiovascular disease, obesity, it's not so straightforward to just get genetics and explain that. So the NIH designed this roadmap consortium to try to epigenotype, if you will, different epigenetic marks across different tissues. And I don't put this up for you to read every single cell type, but what I do want you to notice is that there's stem cells there represented. There's also an adult, and there's also a fetus. And this is a little bit simplified, but you can see that there's the influence of tissue and time, because you have a fetal epigenomes, and you also have adult epigenomes. So there's the time component, and then there's the cell type component as well. So it becomes a very arduous task. It's not too easy to get all these tissues, and particularly even when you go to one single organ, there's different cell types in that organ. So it becomes a little bit difficult, at least to try to get epigenotypes. Even within the brain, there's multiple cells that you have to get these epigenotypes, if you will. So I just want to make the point that there's the time passage here, and there's also the complexity of cells, as I mentioned earlier. So epigenetic modifications, as I mentioned, these are changes in the expression of genes by definition that are non-dependent on the sequence of DNA. One of the things that we do in epidemiology is in human studies is measure methylation because we can do it systematically, at least across people. So oftentimes what we're trying to measure is methylation of cytosines. So you can see there the cytosine molecule. The methyl group is the CH3 group that can be attached to the 5' position of that ring. That's still read as a cytosine, but now it's a 5-methyl cytosine. So the cytosine is not changing the genetic code, it's just that slight tag there. There's also histone modifications, which are the protein complexes where DNA can be wrapped around. And as you can imagine, that can make DNA more accessible or less accessible. So therefore, you can kind of regulate gene expression in that way by blocking or having access to the DNA or the actual gene body. And then finally, there's non-coding RNAs. So these are either long-coding RNAs, microRNAs. These are RNAs that are interfering with that process of transcription. So you're still regulating expression, but it's not at the genetic level. What I would say is that in human studies, very much like genome-wide association studies like the GWAS era that we had in the 2000s, we're still trying to do the EWAS, which are epigenome-wide association studies at the methylation level. There's no systematic investigations of histone modifications just yet in human populations. I mean, there's few small studies in specific cell types, so we haven't gotten there yet. The same thing with the non-coding RNAs, the microRNAs, or the long-coding RNAs. So at some point we'll get there, but I think right now what's ready for prime time and you'll see is methylation of DNA. Okay, so why do we care about methylation of DNA? This is a very simplified view as to why we care. So these lollipops right there next to the X gene, you know, some gene that you care about, the lollipops represent cytosines, and if they're not shaded, they're unmethylated, and they're shaded, they're methylated. In a very simplistic view, one of the things that we realized early during the discovery of methylation of DNA is that if the CPG islands, so these are regions in the genome that are not random, so they're CG repeats throughout the genome, oftentimes they're located upstream of the promoter of the genes. If they're heavily methylated, gene expression is repressed. If they're not methylated, gene expression can be turned on. This is not deterministic. It's not that you're turning on and off genes, but this is a way to do it across multiple cells that you can kind of regulate how strong you want a gene to be expressed, how many transcripts you want. So the whole hypothesis here is because you are changing the expression of DNA, of specific genes, you're changing maybe the chromatin conformation as well, how accessible that DNA might be. The whole hypothesis is that that has an influence in health and development. And that's why we care about, at least. Okay, so I study early life exposures, at least one part of my research programs, and I got into this during my PhD. And one of the reasons I find it fascinating is in the process of fetal development is that epigenetics is very dynamic. As you can see here, on the y-axis is the degree of methylation, on the x-axis is the process of fertilization, all the way from having fully mature sperm and eggs, where the degree of methylation in the genome is higher or fairly high. You know, there's no units on the y-axis. This is just qualitative to show you what's happening. Upon fertilization and implantation, what happens is that the methylation levels get erased and reestablished again. So it used to be thought that methylation gets completely erased and then reestablished. There's very few exceptions to that, which is the purple line that you see there. These are imprinted genes. So you get two copies of a gene, right? You get a copy from the paternal side and a copy from the maternal side. Imprinted genes are genes that are silenced either from the maternal or paternal side, and sometimes both copies need to be silenced, and one copy has to be silenced, but not the other. And when that goes wrong, you get imprinted diseases. So this is a handful of genes. I think it's estimated now maybe a little bit of over 100 genes out of the 20,000 that we have that might be imprinted. So that's a very special case, but for the most part, the great majority of the genome gets demethylated and then remethylated again early during fertilization and implantation. And then the cells are gonna do what they're gonna do. They start, you know, the pluripotent stem cells start dividing into different types of cells, including germ cells. So you have 3 generations here, and I think we argue that this is a very critical window of development, where if you get an exposure, an insult, or a stressor, that's gonna program your epigenome down the line. You know, because we're in environmental health sciences, a lot of us are thinking about risk assessment, right? How we set regulations. You know, pregnant people are one of the vulnerable populations that we take into account. And I think in terms of regulation, if any of you are interested in that aspect, doing this during that, at least getting doses or setting standards based on pregnancy specific outcomes might be best, because you're protecting 3 generations. You're protecting the F0 generation, the F1 generation, and also the sperm cells, the germ cells, which is the F2 generation. So epigenetics plays an important role in tissue differentiation and also fetal development, as shown here with the programming of epigenetics cell differentiation. So how do I think about epigenetics? So I that's blocking the, yeah, let's see if I can do that. If I say, okay, that, let's see.
Try not to hit me.
Okay, so, okay, there we go. Okay, now you can see that. So I think about epigenetics as the gatekeeper of information. DNA is very important. I don't want to make the point that genetics, there's diseases that are highly, that have high disease penetrance from genetics. And we have models to describe those very well. But in general, from a public health perspective, I'm interested in complex diseases, things like metabolic disease, cardiovascular disease, and so on. So we have DNA with all this information. And the analogy that I like to use is that we use all these tags like metal groups, the same way you access information in a library is stored and sorted by sections. And there's also these kind of submarine shelves that kind of move up and down. I don't know, many of you maybe haven't been to a library in a while since we don't use that, but most universities still have these collections, and you can access that information moving from one side to another when you need it, right? But sometimes it was maybe even years before people move one of those shelves to access any given journal, book, or citation. So this provides a concept of biological memory. So this is a window into the past. So I'll try to elaborate on that a little bit. So if something happens to the cells mitotically, that change gets passed on. So you have this biological memory, mitotically speaking, at least in cells. But because you're changing the genome, something is happening to that genomic potential, right? The ability of that gene to be expressed. So this is what we call epigenetic programming, particularly during pregnancy. So I think that provides a snapshot into the future of health and disease. I do think that picture is blurry. It's not as deterministic as genetics, like the BRCA1 gene for breast cancer. I think that provides a little bit of a picture, but it's not a complete picture as epigenetics might be malleable down the road. So there's two concepts here. Is that epigenetic programming can influence genomic potential in terms of transcription, but also provide some sort of biological memory down the line. So probably some of you have seen this. This is a seminal work performed by Dana Dolinoy at Michigan. And what she was able to demonstrate here using the agouti mouse is that exposure to BPA shift the coat color of these mice. So this is exposure during pregnancy to BPA, which is an endocrine disruptor and a plasticizer found in bottles, or used to be found in bottles. Now they replace it with BPS, but that's a whole nother lecture. Kind of created this shift in the coat color of the offspring that became out of this yellow phenotype compared to the pseudo-agouti or the heavily mottled phenotype that is a little bit more brown compared to the yellow coat color. And so this is a prenatal exposure to an endocrine disruptor, BPA, which is completely synthetic, disrupting something so obvious like coat color. So that was pretty fascinating, right? When you talk about triglycerides or blood pressure, that's a little more abstract, but seeing something in a picture makes it more attainable, understandable. What she was able to do is go to the agouti locus and a group of genes within this locus and demonstrate that these box plots show the CpG sites within this gene. And there was a decrease in methylation at this locus that was linked. So this locus controls coat color, so that's why she targeted the specific gene. And she was able to show that animals that were exposed to higher BPA in their diet compared to control animals had lower levels of methylation at those CpG sites. And remember, so methylation of a specific cytosine is a binary event. Either you're methylated or not, but because you're reading across multiple DNA strands, you're getting an average. So this is why the variation comes on. So that's something that sometimes is not very clear to people. Either you're methylated or not methylated at a specific cytosine, but because we have many DNA strands from different cells, there's some uncertainty on that. So that's why you get variation. It's not like the genotypes, or you are either one genotype or another. So the reason this matters is because these yellow mice tend to develop, and she later on went to show that, yeah, BPA definitely leads to this insulin-resistant kind of obesity phenotype in these yellow mouse models. And this is from a prenatal exposure. So that, to me, at least as a graduate student, that was very exciting, or not exciting, but at least eye-opening to see that a prenatal exposure has such a profound effect on later life phenotypes. Okay, so I started thinking, well, I'm an epidemiologist and I do human studies. So how do we do this? We know that there's thousands of chemicals, many of them cross the placenta. We used to think that the placenta is an amazing organ, but It doesn't filter everything out, particularly the small molecules. Many things cross the placenta that reach the internal compartment. Some of these chemicals, once they reach the fetus during pregnancy, can also cross the blood-brain barrier. Things like lead and mercury get into the brain, and this is how neurotoxicity would occur. But really, the question that we're trying to get at is whether that changes epigenetic marks, particularly during development, as the graph that I showed you why epigenetics was important. But, that's scientifically, that's interesting. I see, if you see changes in epigenetics, well, that's great. But usually our NIH colleagues want to know why does it matter? The clinicians, does it matter for newborn health? That's what we care about. Yeah. But like, why do you care if you see a methylation change? That has no relevance to health. So we're trying to relate that to newborn health, things like birth weight, gestational age, neurodevelopment, things that people care about down the line. And the reason why we care about things like newborn health is because that kind of sets up the framework for life course epidemiology. So there's a pretty robust literature from observational studies in epi correlating like things like preterm birth and birth weight to later life disease risk. Again, this is we're just talking about risk. I am missing a pretty important portion in this diagram, and I feel personally responsible because we keep doing maternal and child health studies without taking into account that. So I'm really glad Rick is looking at this because we have all kinds of maternal recommendations, right? When women are about to get pregnant or when they get pregnant, you get bombarded with recommendations. You should eat this fish, not this fish. Make sure you eat fish low in mercury. You should take prenatals. But we have little to no information on that. And I think that's from the lack of data. So I'm really glad Rick is looking at this question that there might be paternal exposures, things that we have known for a long time that are detrimental, things like smoking on the maternal side, that's one place to start. Well, smoking likely, might have some paternal effects on that side as well, and diet and things like that. So I think the lack of recommendations, I have looked into this, probably come from the lack of data. Not to say that that's are not, the paternal side is not important. It's just we have very little data to do science-based recommendations. So I'm glad people like Rick and many of you are looking into this question, which I haven't contributed too much, but we're working out together through a mutual colleague to look at sperm methylation as well from the paternal side. Okay, so this is from a review. If you're interested in thinking about epigenome-wide association studies, particularly as it relates to humans, we set up some recommendations. The reason I'm showing this diagram here is because you have the life stages, and this is a layer of complexity that maybe you don't have with some of the other biomarkers, particularly in genetics, where you have the prenatal period that I show you why this is so important. I didn't show you data for that, but during childhood, the epigenome continues to change, not as much as it did during fetal development. And then during adulthood, there's other sets of problems where there's epigenetic loss, and there's also epigenetic dysregulation. So things are not what they were supposed to be as when you were younger. And this is tightly connected to aging. So there's different life stages that you have to study. So now we have the complexity of cells, we have the complexity of time to study, and then we have the complexity of moving environments too, right? The environment that you were exposed to prenatally compared to when you were born, as you age, right? People change lifestyle habits. It makes it a difficult problem to track, but it is tractable with good study designs. There's genetic variation that influences epigenetic variation as well. So this is an important concept where you have genetic variation from the population, like a SNP that has some minor allele frequency of like 5 or 6%, so meaning 6% of the population might have it. That can actually influence epigenetic variation within that region. So I think this is promising to try to explain complex diseases, right? So we all know people that smoke into their 90s and 100s, and they're doing very well, which is perplexing, right? So why do some people get sick while other people might only smoke for 5 years and develop lung cancer or throat cancer, right? So this is a very interesting problem for to think about. There's genetic susceptibility that might interact with the epigenome to try to explain complex diseases. Here we're interested in allergic disease. So some of these pathways for asthma has been pretty well worked out, at least for some phenotypes of asthma. So in that literature, there's definitely evidence that there's an interplay between genetics, epigenetics, and environmental exposures. Things like air pollution that trigger both asthma initiation and exacerbation as well. So this is what we do in our group. We get DNA from different sources, and I want to make the point that there's different sources. We're using microarray technology, which I think some of you are also implementing, very similar to genotypes. These are microarrays as well if you're not doing exome sequencing. That measures getting close to 1 million CpGs per sample. And I think there's about 24 million, the estimate of CpGs across the human genome. So we're only measuring a fraction of those that are fairly representative. So you might have a CpG island with 30 or 40 CpGs. The array might cover only a couple on that specific region. But so still the coverage is good, but it's not as good as getting at the entire region. And then there's a set of bioinformatic analysis that you have to do down the line. And there's a lot of work to do in this area, both in the bioinformatics front, but also in the statistical front because of the issues of small sample sizes and very high degree of omic features. In this case, similar to GWAS, we started with studies in the hundreds. Now we're getting with studies in the thousands of people. And, hopefully we'll work up to something like the UK Biobank where you have hundreds of thousands of individuals and hopefully have more power to statistical power to get signals out of how much testing you're doing, right? Because the p-value is no longer 0.05. The p-value is 10 to the negative 8, 10 to the negative 9 that you're looking at. Okay, so I'll give you an example of that. So for this talk, I tried to concentrate on general projects, given the audience has a wide variety of interests. But I'll start with an epigenome-wide association study or a couple that kind of showcase these concepts that I introduced. So my epidemiology colleagues like to say that this is a fishing expedition, and sometimes this doesn't review very well for NIH, right? Because basically I'm just throwing a net and seeing what falls out, right? The hypothesis is that there's an exposure that influences epigenome, but that's a very non-specific hypothesis, right? There's 20,000 human genes and which genes are being influenced. I have no a priori hypothesis. I'm just saying there's some influence on the epigenome, which is very non-specific. But nonetheless, I think we can get a lot of information. So this is a birth cohort. As an example, we looked at smoking during pregnancy and placental methylation. So smoking during pregnancy is one of the biggest risk factors for perinatal outcomes that are not, or bad perinatal outcomes. It causes low birth weight, so I think no one will debate me on this, that smoking is a causative factor for low birth weight and reduces fetal growth. That has been established over and over and over. The prevalence of smoking varies a lot. So, where I'm from in California, our prevalence is fairly low. I think last time I looked up a statistic, there's 2020, I think we're down to we're getting very close to 1% of pregnancy smoke, which is super low. But then you have states like West Virginia that almost a quarter of the pregnancy smoked during pregnancy. So there's a lot of variation. And then if you go to Europe, some countries have even a higher prevalence of smoking. So then again, when you are working with epigenetics, the tissue really matters. As we like to call it, this is the tissue issue, right? Because you have to take into account what tissue you're looking at, but this also provides an opportunity. So here, the opportunity is to study an organ that is highly relevant to some of these outcomes, which is the placenta. It regulates nutrient exchange, waste exchange, and it also has metabolic functions. So it's tightly intricated in fetal growth and produces hormones, and it also has receptor for hormones. So it makes sense to study placenta when you're studying fetal growth. So we use a prospective birth cohort. So this is when expecting people are recruited during pregnancy. We saw them in the first trimester of pregnancy, and then we collected placentas at delivery. This is a study for 141 mother-child pairs. And then in the table, you can see there the descriptors of the study. This is in Canada, so all the participants were Caucasian, but what you can see is that the smoking prevalence was about 8 or getting close to 9%, so only 38 smokers out of the 441 reported smoking during pregnancy. And we also had some biomarkers of smoking and validated this self-reported smoking early during pregnancy. This is during the first trimester. All the deliveries were term deliveries by design. We did not focus on preterm deliveries. So the birth weight was within the normal range. So this is what essentially we did on our hypothesis in a diagram. So you have smoking during the first trimester of pregnancy. We proposed that methylation of the placenta was a mediator, so was on the causal pathway to lower birth weight. There is a main effect of smoking, so we were able to verify that. So we got 175 grams, on average, babies were 175 grams smaller of moms that smoke compared to babies of moms that didn't smoke during pregnancy. So that was a main effect, nicely replicated from the literature. And then we asked the question, well, is this related to, so we did two things, and it's a little bit hard to, there we go, I can use the mouse maybe. In this side of the equation, we were looking at the question whether smoking causes lower birth weight, leads to changes in the epigenome of the placenta, and whether changes in the epigenome of the placenta leads to lower birth weight. The take-home message is the signal from the placental methylation to birth weight was fairly low. I think we only got maybe a handful of hits that passed statistical testing adjustment, but the signal for smoking, which is in this side, was very high. You can see this is a Manhattan plot with the human chromosomes here annotated by position, and this is the negative log of the p-value. So we had effect sizes that were extremely large with a p-value 10 to the negative 30. So here you have a main effect, and then we ask the question whether there's mediation through placenta, so causal mediation through the placental methylation. What we were able to show here is that among moms that smoke during pregnancy, the indirect effect was quite large and mediated through the placenta. And then for moms that didn't smoke during pregnancy, at least those hits, placenta is really not, those p-values are not driving that association. So the take-home message here is that there appears to be kind of like an exposure mediator interaction here where the placenta is really not the driver of lower birth weight in this case, at least in this subsample. We might be underpowered to detect associations, but among term babies that were normal of the normal weight range, smoking really causes low birth weight, and this is initiated by smoking. So smoking initiates that causative action for the lower birth weight, looking at placental methylation. We use a statistical tool to decompose the effects, and that's why you see smokers and non-smokers. But this is just a proof of principle. We know smoking is bad. We had the main effect, but at least biologically, you can explain some of these. The one gene that I'm interested in here that I didn't know anything about was the PBX1 gene. So this is the second row, that CPG. Had one of the highest effect sizes right here. Kind of hard to see that, but I think it was, 300 grams lower birth weight mediated through that pathway. So that effect size was even larger than the main effect size that we obtained. Of course, this is just an estimate. There were a degree of error around that. So we are in the higher end of that confidence. But PBX1 regulates osteogenesis of bone formation, which makes sense because when babies are born, they don't really have that much fat. They're mostly, their weight might come from the bones. So this is really interesting. Now we have mechanistically showing that maybe this gene, which is involved in osteogenesis or new bone formation, is influencing birth weight through smoking, initiating that pathway at least. And if you're in epi, we propose this exposure mediator interaction to be highly relevant for epigenetic epidemiology, where the mediator itself for the epigenetics might not be a driver of that causative factor, but you need exposure in order to initiate that. So I wanted to also show you, I think some of you are working with metals. We're also interested in prenatal metal exposure, things like lead and mercury that have been connected to poor neurodevelopment. And I'm not presenting those data, but this is a cohort in which we've been able to show that metal mixtures influences neurodevelopment detrimentally. And there's also essential metals that might counteract some of that effect. So we've been after biological explanations similar to the smoking project to see whether there's the epigenome might explain some of these observed associations. So this is a prenatal cohort, very similar to the previous one, but this is a Boston-based cohort. We measure about 12 metals in erythrocytes, in red blood cells, quantified by HPLC. And you can see here in blue, they're the nutritious metals or the essential elements, things like copper, selenium, and zinc. But we also have toxic metals or metals that are not essential, things like lead, cadmium, and arsenic. So these metals are a nice case study because they kind of do opposite things. Some of the essential metals reduce oxidative stress, and some of the non-essential metals actually increase oxidative stress. Both, that's just an example, both a factor for aberrant methylation changes. So then we looked at methylation. We looked at methylation in cord blood, and then something else that we've been trying to test is whether if we see changes in the cord blood, so this is prenatal exposure, and then measuring epigenetics in the cord blood, whether these changes actually persist as children start to develop. So that's something that we've been testing over time, looking at methylation changes at 7, 9 years, and now we're getting samples at 14 years of age in the blood. So we're still studying leukocytes. And the leukocytes are turning over, but if you remember that epigenetics are methodically passed from one cell to another. So that's the hypothesis that if something is aberrant, if something is associated with a metal exposure early during development, that change will be propagated through mitosis at least. So these are genomic plots. The dots are individual CPGs, and then we put confidence intervals about that, corrected for multiple testing. These are different genes. And the reason why I'm showing you this is, for example, this is a nice example with effect sizes going the opposite direction. This is manganese and magnesium. So this is a non-essential and an essential metal going in opposite direction in a gene involved in immune development and some genes involved in neurodevelopment as well. So we had several examples where we identified entire regions where things were going in the opposite direction, at least here for an essential and non-essential metal. This is one of the examples. And we also saw some sex-specific effects that kind of track with the neurodevelopmental effects that have been reported in the literature as well for sex-specific effects among males and females. So I'm not going to go into a methodology of this, but this is just to showcase persistent changes that might remain after a prenatal exposure. The same way, for example, BPA influenced coat color, we're really interested to see if a prenatal exposure in humans will persistently influence methylation of DNA across time, persistently at least. And I think this is an outstanding question in the field, whether things persist or not. I was having a conversation with one of you today about that in terms of persistence. And I think what we've been able to document, even with the limitations of epidemiological studies where you're not able to follow every single individual after 8, 10 years, or even 14 years where we're seeing the kids again, is that about 10% of the changes that we see in cord blood kind of persist after time. And we have seen this, we have a study looking at phenols, and we have this study looking at metals where we have repeated exposures, and not every single change persists. So this is also hopeful because, not everything is programmed during the in utero phase, or there might be other influences like nutritional and lifestyle influences that might be able to offset this. And that's something that we're trying to pursue now. I'm just gonna keep an eye on time. So for the last portion of the talk, I wanna talk about epigenetic aging biomarkers, and this is a new development in the field. Has generated a lot of excitement, I think, and so I wanted to show you some of those data as well. So this concept of epigenetic age or DNA methylation age is fairly new. For the longest time, as you know, the scientific community is interested in trying to develop a biomarker of aging. Probably a lot of you heard about telomere length, right? So telomere length, the telomeres get shorter as you age. That has been well documented. But if you tell me your telomere length, I have no idea what your age is, right? I just know it's relatively speaking should be shorter as you age, but there's no way to kind of guess how old you are if you give me your telomere length. So the community was interested in developing a biomarker to try to basically predict someone's age based on biomarkers. So this concept of a biological epigenetic aging clock has been pursued by several people. Very early on, there were changes in the epigenome that were linked to age. So if you just look at age and epigenetics, it appears that there's very, very strong changes in the epigenome related to age. So with the aid of some statistical tools, this is a colleague used to be at UCLA. Now I'll talk a little bit about that. He moved to a private enterprise at Altos Lab, but he developed this epigenetic clock, Steve Horvath, using methylation changes of many genes. Interestingly enough, he did this completely with publicly available data. I would say this is one of the really most exciting developments in the aging field. So you can literally run a research program with publicly available data nowadays. So the bioinformatics is an exciting field to be in with the wealth of data that we have. But in that plot, what you can see here is that the numbers indicate different studies and the colors, different tissues, I believe. So different tissues and different studies track very well where he trained a model to predict age. So here's on the x-axis MH, so it's methylation age, and this is the actual age of the participant with an error rate of 3 years. So I don't know about you, in epidemiology, we don't see correlations of 0.9. So that's very, very exciting. I mean, even the telomere length correlations that I've seen are 0.5, 0.6 at best, with a large degree of error. But here we have a nice biomarker of someone's age. This is being used in forensics. So now it's pretty useful to be able to tell the age of a perpetrator, right, of a crime. Is being used by the aging community to track interventions as well, because as you can imagine, if you decide a human study to test pharmacological intervention, you don't have the time to wait until people die or get sick. Might be decades before any effect is shown. So having a biomarker is very useful. What we're really interested in, at least from the point of view of aging, is that we know that this works fairly well. So, right, so there's a pretty good correlation, but there's bumps, right? And you can see that there's a tighter, the data is a little bit tighter here, 0 to 20, and then the data becomes a little more noisy. So the whole concept is that higher levels of epigenetic aging, so when this is the one-to-one line, what you would expect that was a perfect prediction. There's bumps. We're basically modeling the residuals or the bumps in the data to see why some people are epigenetically older and why some people are epigenetically younger, independent of chronological age. So besides forensic applications, it's not very useful to know how old you are, because you know exactly how many days old you are. Everyone knows their birthday, and you can probably get it down to the hour if you wish so. So it's not very useful, but it's useful in the sense of having a biomarker of biological aging. Besides the forensic application. And what was really exciting about this clock was that it was multi-tissue. For most human tissues, it worked okay. And this is both tissues like brain, liver, but things like sperm, it didn't work very well in sperm, and it didn't work very well in breast tissue. So there's hypotheses as to why that might be, but those are the tissues that didn't perform as well as other tissues. And some tissues perform better than others. In blood, the clocks work fairly well. So now there has been an explosion in the field, multiple clocks. Everyone is developing their own clock. There's different goals of developing a clock. For example, if you're trying to predict gestational age, which some of you in perinatal epidemiology know that that's a very difficult task to do, to predict gestational age accurately. There's multi-tissue clocks, which are exciting, right? Because if you're studying cardiovascular disease in humans, very difficult to get a cardiac sample in a healthy individual. So you want to have something like blood to try to reflect cardiovascular disease. Or you're trying to study brain aging, very difficult to get a sample in a healthy human. So it will be useful to have a proxy. And in this case, this is why the multi-tissue clocks are exciting. There is pediatric clocks designed for children. And then again, the clocks that are not multi-tissue, oftentimes what they do is that they increase they increase the prediction accuracy. So there's clocks, if you use a clock that is just purely based in blood, you might get an error rate of about 1.5 years. So it's more accurate at predicting chronological age. But oftentimes people are not interested in that because everyone knows their age unless you're using it for forensic applications. And then finally, the last section there that says second generation clocks. It's very interesting. So people are actually integrating other sources of data, things like CRP, things like lipids, to integrate it with epigenetic data to try to predict morbidity-specific and mortality-specific clocks. So there's clocks to try to predict cancer. There's a whole company around that called GRAIL that is trying to predict cancer many years before cancer initiation by integrating different biomarkers beyond epigenetic clocks by using epigenetics to track age along with these changes in other biomarkers. So different populations, different endpoints, generating a lot of excitement in the field. So I wanted to show you some data. Again, this is a different bird cohort. This is in the Salinas Valley in California, where there's a lot high agricultural activities called the Chamacos cohort. It was based mostly in Mexican-American farm workers that are working out in the fields, collecting fruits and vegetables. This is a gestational age clock based on cord blood. You can see that it doesn't perform as well as the adult clock, but you're getting, these are two different methods, Booleans and Knights, based on Illumina data, but we're getting a correlation of about 0.7 to predict gestational age. And then we have these outliers too that bring the question, whether what's happening here for individuals. It might be something to do with the delivery, and we have clocks that tend to be a little more noisy. So the quality does vary, and you have to kind of really develop expertise in terms of how the clock was trained and what is actually measuring. This is part of the task that we're trying to do. So I had this student who was really interested in programming based on metabolic parameters during pregnancy. So we know things like high glucose levels during pregnancy are detrimental to both mom and the fetus as well, high lipid levels. What she was able to show with the more accurate clock here, you can see that the estimates are better. Things like higher triglycerides, higher lipids lead to decreases in epigenetic aging, acceleration at birth. Deacceleration used to be seen as a good thing, at least in adults, and we have started to look at children, and we're finding that a lot of the toxic exposures are actually deaccelerating the clock, which might not be good for health and development. If you think about a fetus, it's a good thing to have normal development. And this was reinforced by looking at preterm delivery, which is one of the biggest risk factors for infant mortality. These individuals had decreased epigenetic aging. So these are point estimates and confidence intervals. But things like parity and infant sex, at least infant sex had different associations. This has been reported that at least males have higher risk of both deacceleration during childhood, but also acceleration during adulthood compared to females. So here's prenatal exposures on the maternal side, things like lipids, preterm delivery, at delivery, parity, having an influence on aging at birth, at least as measured by this epigenetic clock. So we're trying to now relate these changes to things like neurodevelopment and obesity risk later in life. That has been related in adults, but not in kids. So this is from the same study. We're also studying social exposures. I became really interested in OP pesticides because that was the original study was designed to look at OP pesticides and neurodevelopment, which show a detrimental effect of the OP pesticides in neuro. That mechanism has been pretty well worked out now. But we wanted to test whether OP pesticides accelerate epigenetic aging. And as I mentioned, we have followed these children. We have cord blood. We have age 7, 9, and 14 years of age blood samples, so we can look at trajectories as well. What we were able to document is the maternal occupation, particularly moms that did ag work during pregnancy. And this is we're talking in the first and second trimester. And then most of the moms had to, because of the nature of the physical work, had to quit during the third trimester of pregnancy, had consistent accelerated aging. So as measured by different clocks. So this is agricultural work and accelerating epigenetic aging. And the 3 different rows that you see there are different adjustments that we did for pesticide exposure. So we adjusted for urinary pesticide exposure that was completely independent. And we also used GIS estimates of pesticide use in the area. And we could not adjust out the signal. So there's something about the physical work, could be also psychosocial pressures as well as economic. We adjusted for SES, male earning the partner's incomes as well, and this association was not explained by that. So there might be something on the nature of the physical work that leads to this acceleration of aging. And remember, this is prenatal ag work in the moms, accelerating aging throughout 14 years of age, so many decades after. And these biomarkers have been linked to mortality and morbidity in adults. It's still a little bit unclear what it means to be accelerated early during childhood or deaccelerated at birth. But you can guess, my guess is that we'll probably see the same patterns once we have longitudinal data in terms of risk factors for cardiometabolic disease and other diseases as well. I wanted to show you another example from a unique experiment or natural experiment that happened in Chile. So this is an area in Chile where individuals were exposed to very high levels of arsenic. So Chile is very arid. They rely on drinking water from rivers that come from the mountains. And this whole town was extracting water from a source. You can see here in this bar plot the levels of arsenic as estimated from the records that they have from the public water company. The levels were fairly low, below 10, which, that was considered safe a while ago. But then they switched the water from another source because of the demand. And the problem is that they did not test that water. The naturally occurring arsenic is a problem from hard water or mineral deposits. the arsenic levels were extremely high, I think reaching 800 micrograms per liter. Just to give you an idea, I think the regulatory standard right now, the WHO standard is 10. So there was a period of time where exposures were very, very high, and then they realized about the problem, and they switch again. They actually put a water treatment plan, and the exposure levels went down to 10 again. So you have this cohort of individuals that were exposed to really high levels. So what the investigator did, this is part of the Berkeley Superfund, they went back, recruited people that were exposed prenatally, so moms that were pregnant during that period of time, and follow the children. So this is a very high exposure during the pregnancy period, and then switching, then you have a comparison group before and after because they switch to a clean source of water. So here we have arsenic exposure, and the reason I go all the way to childhood is because there's an error of about a year to maybe year and a half. So most of the exposure occurred during pregnancy. We don't have the specificity to say just on the 9 months, but could have been early during life as well. And then we recruited people back when they were in their, the median age was 50. So this is decades later after they were exposed to really high levels. And basically we have looked at epigenetic marks and epigenetic, I'm really interested in epigenetic aging, and that's what we looked at. So the exposed group on average, or the median level was, the exposure was 555 micrograms per liter. This is a very, very high dose. And the unexposed group was fairly low, below the 5 micrograms per liter that we use in the US now. So this is a postdoc in the group. She was able to document in a pilot study. These are, I believe, 20 exposed and 20 unexposed individuals that there was acceleration of multiple clocks here as shown on the y-axis. And the effect sizes were about 5 years, at least for the clock that showed the highest acceleration. And what I was really interested in this pheno-H acceleration. So the pheno clock is a marker for, a good biomarker for multiple morbidities and highly tied to mortality as well. In this cohort, cancer incidence has been shown to be very high, particularly lung cancer and bladder cancer for the exposed individuals. So it's a surprising, probably not, but it's nice to show a molecular marker being affected, a non-molecular marker related to mortality and morbidity that goes along with epidemiology showing increased incidence in cancer on the highly exposed individuals. And remember, this is from a prenatal exposure. So we've been trying to convince National Institute of Aging that studying pre-pregnancy or, pregnancy exposures is very, very important when you're thinking about aging. And I think they're beginning to appreciate the concept that if you want to do prevention, you've got to start very early on. Aging has been historically studied among older adults, which I think there has to be work in that area. But the National Institute of Aging, if they're thinking about prevention, they got to start early during conception. And I think they were receptive to this idea because they have funded part of this project too. So whenever I present to toxicologists or individuals that are doing experimental work, they're like, well, this is all epidemiological, there's, what about confounding? And there's many factors. So we collaborated with colleagues from the EPA. They performed a randomized controlled trial of diesel exposure. So these were 13 individuals that were exposed to both diesel exhaust and clean air in these chambers at the EPA. So they were controlling exactly how much diesel particles were coming into the chamber. And then during the clean air exposure, they had filters that were filtering even outdoor air to a very low concentration or particles that were non-detectable. So we got lung tissues from these individuals. So yes, adults consented to getting a lung sample. So they got a bronchoscopy and they got a lung sample out of them. And we did methylation of these samples. And we were able to show multiple effects across the genome. So this is a genomic circular plot showing where the effects are happening across the genome. And then I'm not going to go through the genes, but the enrichment analysis show this EGF and its receptor to be differentially methylated, which has been known to be linked to lung cancer. So now we have a molecular mechanism potentially linking ambient exposure, things like diesel exhaust that we know is a non-human carcinogen, differentially methylated in the lung of participants that were exposed. So this is a randomized controlled trials, a crossover trial, so people were exposed to diesel exhaust and clean air in different occasions with a washout period of a few weeks as well. So we have causal evidence from randomized controlled trials, at least in humans. Very difficult to do now. They halted these experiments for a while because someone had AFib in one of the chambers, but I think they're ongoing again. We show age acceleration for diesel and ozone as well. But I want to move a little bit faster and end with the last thing, which is that we're trying to develop biomarkers out of the epigenetic data. This is specifically looking at polygraminar PBBs, trying to develop a predictor. Here you see methylation data, trying to predict PBB concentrations. In the training data, we were getting correlations of about 0.8. But whenever we went to an independent data set, looking at testing, our correlations dropped to about 0.3. I think the highest that we got, was around 0.3, 0.4. So this is just to showcase that methylation is liable and might serve as a biomarker of exposure in the long term. So with that, I do want to leave some time for questions. I just want to say that, epigenetics is definitely being used. This concept of epigenetic age is being used by industry. Here's an insurance company trying to risk stratify people based on their epigenetic age. So I think that this is definitely a misuse of our biomarkers, and probably not to protect public health, but instead, to benefit insurance profits, which is not great. And I had a few more slides about this new enterprise. Jeff Bezos funded Altos Lab. I think they got a $3 billion investment to try to cheat aging. They recruited a bunch of people in academia, but there's a lot of enthusiasm in the tech sector to try to develop interventions for aging. And we still have an issue of diversity. In genomics, this is well known for genetics, even worse for epigenetics, but most of the samples that we're getting are from Caucasian populations. So how generalizable these things are is still remains to be determined. So I think I said all of this. I just want to take a moment to appreciate, my team, my collaborators as well. A lot of these cohort studies have been going on for 20 years, which I definitely did not start, but they have done all the hard work for over 20 years to follow these individuals over time. NIHS, as well as the P42 Center at UC Berkeley, and my team that has done much of this work. So with that, I really want to, say to the students, I am recruiting postdocs, so feel free to reach out. I think this is the link to the application, but I'm happy to chat one-on-one as well. And with that, I would love to have some questions and discussions. So thank you. Yeah.
Wonderful talk. I'm interested in preeclampsia. You're interested in smoking and birth weight type things. I'm wondering if you have any studies behind that.
So in that metal cohort that I showed you, we do have about, I think we measure metals, essential and non-essential, in about 1,200 pregnancies, and the preeclampsia rate was about 4%. So the cases are a little bit low, but we have tested this. We have a paper under review looking at metals, and it particularly appears that copper, surprisingly, and there's some animal evidence that copper decreases preeclampsia risk. So it's still under review, it's preliminary, but that's what we have looked on from epidemiology, whether non-essential or essential metals decrease or increase preeclampsia risk. Yeah.
Have you seen anything that changes the relationship to smoking? If smoking has to affect that birth rate?
No, but that's a good question. Yeah, as an effect modifier, we have control for it as a confounder, as is one of the major drivers, but not as an effect modifier. But that's a good point. Yeah, that's great. And our cohort, thankfully, is higher SES, highly educated. This is the Boston-based cohort. The smoking rates are really low. I think we had like a total of 12 smokers. So we might not have the power to look at this, but even with those numbers, I think it's a good idea to run that.
If you do, take a look at normal parity.
Okay, already. Okay, that's good. Thanks. Yeah, appreciate it. Yeah.
So I know you probably can't start because you had 38 smokers in your first group to not do this, but I was just wondering if you divided up the smokers into like the severity of smoking, whether they were like mild, kind of moderate, or severe, and see if you were able to get more like the percent of.
That's a great suggestion, and we did not. We did have biomarkers of cotinine, so I have to go back and see what the levels were. But we basically, I think with the cutoffs that are suggested, we did reclassify everyone that reported being a smoker as a smoker. So we didn't have a misclassification problem, but we did not look at concentration dose response. And that's a great question to go back and maybe look at that. Yeah, I think part of it, the issue is like, the variation wasn't much, so how much we would gain from it, but that's still a great point, yeah.
You mentioned that discovery experiments don't go over so well in some studies actually.
Mhm.
Wondering if
Yeah.
you're trying to quantify
Yeah.
kind of discovery, but doing a meticulous study
Yeah.
design on normal.
Mhm. Yeah, that's a great point, and I feel for, I don't know, this is my personal bias. Study section always gets excited about new things, right? So doing something boring like quantifying normal might not be as exciting for study section as it should be, but yeah. I'm sorry, I think I just had male and I had a proportion of males, but they were males and female offspring, yeah. I'm sorry, I think I just had a line that said male and I just put a proportion of males, yeah. But they were males and females, yeah.
So you mentioned smoking really low, which is truly common, but
Mhm.
in our Michigan ethno cohort, we found that over 50% of the moms use marijuana. That's something.
Yeah.
That's just taking off.
Sure enough. Yeah, and I'm really interested. If anyone wants to do a cannabis study, I think it's going to be a public health problem in the future. I mean, California legalized it a while ago, and I can say just from observational data, it seems like more people are using cannabis than actually. So like you don't see anyone smoking in California, but you see plenty of people smoking marijuana or edibles, or the stores are everywhere. And then the tobacco stores are almost non-existent. It's really hard, to see smokers, like tobacco smokers. So yeah, that's a great point, and we haven't looked at it. Take it, these cohorts were, most of them, well, the Canadian cohort was in 2015, the other one's 2000, the Chamacos cohort was, so this was a while back, so maybe it was low, but I think contemporary use probably is very high. Yeah. Right. But, I think OIHA, the Office of Cali, PA, did a risk assessment for this, and part of it was that a lot of people were using edibles for morning sickness, so they're not smoking it, and there's this conception that because it's natural, it's good, and it does help morning sickness. I mean, there's some data showing that, so it's very interesting. Like, it's not, it's not indicated, but people are, of course, going to use it off-label.
Keep an open mind, it might be beneficial.
Yeah, no, of course. Yeah, I have no argument against it. I just think, the endocannabinoid system is highly active during fetal development, and so there's a lot of receptors that you're gonna get the cannabinoid. So yeah.
Any questions online?
Yes, I have 2 questions.
I'll go ahead.
Okay, so well, enjoy your talk. So the first question is, have you noticed of the epigenome instability commonly induced by different the chemical? So, and how you can measure the epigenetic instability?
So the stability over time, you mean, or across tissues?
Yeah, overall, for example, the epigenome become unstable because you actually focus on the big peaks, but they also have. Do you notice that the overall noise is increased, and which actually have a clinical impact?
Yeah, I haven't looked at this question specifically. I can tell you from our studies looking at smoking that, about 7,000 from the meta-analysis, we know out of the 20,000, approximate human genes, about 7,000 change with smoking. There were methylation changes related to smoking. Most of these got reverted back after quitting, at least from the cohorts that have quitting information and pack years use. So I think, at least qualitatively, what I can say, I think a lot, at least with smoking, most of these might be reversible over time, while some of these, like the HRR, is pretty permanent. I mean, not permanent, but it's so liable that you can tell in terms of pack years how long someone smoked by looking at methylation. With some degree of accuracy, of course. So I would say, from smoking and leukocyte methylation, what we know is that most of the changes kind of revert back after people quit, but not all of them. And to your second question about relevant to clinical outcomes, now people have looked at HR methylation because this iron hydrocarbon repressor receptor, methylation of that gene has been linked to cardiovascular disease independent of smoking. So I think it has potential for clinical biomarker development. And I keep going back to smoking because that's where we have most of the data from people that cohorts that already collected that.
Sure, sure. So one more question. So now you have so much data about the epigenomic, the whole genome behavior. So when you compare this with previously published, GWAS study, does this data make sense in terms of how nicely they collaborate with each other, or some actually the conflict with each other? So what's your feeling about the future for integration of the data sets?
Yeah, the MQTL, so the methylation quantitative trip loci, this is where a SNP influences methylation. I think that's very promising, and we're gonna have to do more integration of that. But as you can, when you layer three things, genetics, epigenetics, and exposure, there's like a three-way interaction statistically, and that you need a lot of samples for that. So I think we're kind of restricted by sample sizes, but now very large cohorts like the All of Us program, I think they're running epigenetics. There's the Million Veteran Project, which is part of the Veteran Affairs federal initiative. They're recruiting 1 million veterans. I know they're doing epigenetics in about 100,000 of them. So I think, and they have whole genome sequences. So I think this will become possible with programs like that, like All of Us and the MVP cohort as well.
Thank you.
Yeah, thank you.
Alan, did you have one question?
Yeah, two quick questions. One is, in the epigenetic changes that are part of the methylation aging marker, have any of those been like detectable at the gene expression or protein level, or is it just too generalized and take that out?
Yeah, someone in the audience might have a better answer for that. I'm trying to think of any data that I've seen. I know there's like RNA-seq clocks now, right? And there's proteomic clocks, but whether there's, what I can tell you from the multiple epigenetic clocks is that the genes are not the same. You train a clock to predict mortality, and then you train a clock to predict chronological age. They do a very good job at predicting chronological age, but if the goal is to predict mortality, the set of genes tend to be different. And the pathways always don't make the same sense. I think there's technical issues, right? Like, so the coverage is, most of these clocks have been developed with array technology. So there's that level of bias already that the arrays are not covering the whole genome. And then there's also the challenge that I think the epigenome really changes with age, like globally. I think there's a lot of changes throughout the human epigenome, and we're just picking and choosing depending on how we train things. But yeah, some clocks are designed to train mortality. They still get a correlation of 0.8, 0.9 with chronological age, and they're still predicting mortality, but they were initially designed to predict mortality. So it's really interesting that things are not completely overlapping, even when the aging biomarkers do a good job of predicting age. But the pathways or genes are not like, even when you looked at them, it's not like, oh my God, yeah, this pathway is related to aging. It's not very clear.
My second question was, have you looked at 5-hydroxy mechanisms?
We did in cord blood. It was fairly low globally. So we did a global assay for high hydroxymethylcytosine. I think it was like less than 1% globally, across the whole epigenome measured by, I think what's HPLC. So I know in neurons is a little bit higher hydroxymethylation, but in blood, I don't, I think, it's not highly detectable. So trying to do epigenome-wide hydroxymethylation can be, it can be implicated in disease, but I think it becomes really hard to detect it systematically the same loci because it's so low. But with sequencing, that would change. I think we were talking about nanopore earlier, and there's a lot of promises about getting sequences, getting methylation, and getting hydroxymethylation on the same sample with nanopore sequencing. But in general, it's very low in blood. I think it was less than 1% globally.
That's it. Let's keep another.
Thank you. Yeah.